I will start now with a short introduction. So basically, in marker-assisted selection, we use a limited number of DNA markers to select for our key target traits in the breeding program without directly phenotyping in our segregating material. And most often, this methodology is integrated within the phenotypic selection to enhance the breeding outcome. Basically, said it very short, we can increase the genetic gain, we can improve the accuracy of selection, and in some cases, we can gain time and have a earlier and much faster selection. The type of markers are mostly DNA markers, and they have two major categories. Markers which are binary, so presence, absence, and markers such as the SNP markers, which are codominant, where, or like a CASPA, say, for a SNP marker, where we can separate the homozygote state from the heterozygote state. When thinking in a practical setting whether to use a marker-assisted selection in a breeding program, one should ask at first place, what can I gain compared to the phenotyping? And generally, marker-assisted selection is more efficient for trait with low heritability. As soon as the heritability is very high, then that is a trait which is just possible to easily phenotype. And the higher the proportion of the genetic variance, which is explained by the markers that we have that are available or that we can use, the more effective marker-assisted selection is compared to phenotypic selection. The reflection that has to be done, it's in terms of time and cost. So when there is a trait which is either very easy or very cheap to phenotype, then most likely we are fine with phenotyping. Nevertheless, we very often have that the key traits of interest for making our varieties better than the the currently available varieties are traits for which is actually maybe difficult or very costly to do the phenotyping. An example in which marker-assisted selection has been implemented since a very long time and in a very successful way are, for example, the gene for gene type of resistance. So I have a disease, I have a gene which really determines then the resistance of that genotype, and by having a marker that targets the positive allele for this gene, I can very easily select without the need, for example, to have a very complicated field trial where the plants are exposed to the disease. I can also do it by doing it in the greenhouse or even in the field, but without the presence of the high pressure of the disease. Another case, for example, is for a trait which might be maybe in some terms, maybe it is with high heritability, for example, the alkaloid content in white lupin, but it might be extremely expensive to do the phenotyping. In our case, one needs gas chromatography, which is about 200 to 300 euro per sample. So in this case, if we have a marker for the selection, it really reduces the cost. And another case that it's especially for the perennial crops, it's in cases where the trait we are interested in is a trait which appears only in the adult plant. So if we are able to have a marker instead of waiting for the months or the years to get the adult plant, then we can bring in the field only pre-selected genotypes and speed up the selection very efficiently. The other question, especially for the ones of you who have followed the unit on marker-assisted selection this morning, is in which case one can use the methodology of marker-assisted selection in comparison with genomic selection. Michael in the morning has already pointed out to the key aspect here, which is that if we have few major genes that determine the trait of interest, then we can have a good success with marker-assisted selection, while genotypic selection is much more useful when we consider very complex traits which are controlled by many genes with small effects. In the morning as well, you have seen from the presentation of Neus about GWAS, the first step of marker trait association discovery. Yet, this is, I hope you will be able to follow the cursor on the screen. This is only the first step from the marker trait association to the actual use in a practical breeding program. So let's say in the first step, what we have seen in the morning with the GWAS, but that can also be the more traditional QTL mapping, can be RNA-seq, can be selective sweep analysis, any methodology that allows you to define gene of interest or marker trait association. After we have this step, ideally, we do confirm these QTLs, these marker trait associations in other environments, in other genetic background, in order to be sure that they are useful for marker-assisted selection. We then really select a few markers to use. We develop according to the type of marker, the assay that we are going to do in the laboratory in order to do the actual genotyping. We do apply these markers for a first time on our segregating material. And we do a field validation to effectively see how accurate and effective is the selection. And only after these steps, then the year after, we can just use the step of marker-assisted selection as a routine step in our breeding. I want to recapitulate this concept with a practical example which was just published on the TAG journal. Here the crop of interest is potato and the trait is resistance to nematodes. In this study, they have picked QTLs from the literature already published. They confirmed via a new GWAS in a new panel of accessions this QTL in a new phenotyping and genotyping study with a new a panel of accessions, and they developed useful markers. In their case, they didn't go for a single market to target one locus, but they decided to use haplotypes of markers, so a set of markers together. And after that, they could convert, in their case, are SNPs markers, so they could convert the SNP markers of interest in an assay. In their case, they decided for the PACE assay, which is basically the same of a CASP assay with a qPCR allows to identify the two homozygote alleles and also the heterozygote plants. And they have applied to a diversity panel, to a test panel, these markers in order to validate their usefulness for selecting the breeding lines resistant to these nematodes. And after all this step, now they are basically ready to use it in a routinely type of manner. For some of you who come maybe from the organic breeding perspective, you wonder if and how molecular breeding methodologies are used or accepted by the organic movement. So basically, in terms of the ethical criteria of organic plant breeding, we have that the cell is respected as individual entity, so no technical or physical intervention in organic breeding is made below the cell level. This means no use of GMO or NGTs type of techniques. Nevertheless, every molecular breeding technique like marker-assisted selection or genomic selection, which are diagnostic types of techniques, they are suitable for organic plant breeding approaches. The other point, which is very important in organic breeding, is that all the steps are conducted in the organic conditions, in the sense that the importance of selecting the plants in the direct environment in which it will be in which they will be cultivated is given a very high importance. And because of this, the molecular breeding approaches in organic breeding are used as complementary tool, and they do not substitute the need and importance of phenotypic selection in organic farming conditions. With this introduction, I would give now the word to Barbara to continue with the very interesting case from Slovenia.
Okay, good afternoon to everybody from my side as well. So thank you, Teresa, for a very nice and informative introduction of the marker-assisted selection. So now we will move to the application case in a common bean breeding program, which is done at Agricultural Institute of Slovenia. This is common bean breeding program is a public breeding program, national breeding program, a small one, but very productive one, I would say. And also in our case, we are using marker-assisted selection as well in our breeding process. So first of all, I would like you to introduce you into the common bean itself. So for those who are maybe not so familiar with common bean, so common bean is one of the most important edible legumes for direct human consumption in the world, and it is a valuable source of protein, carbohydrates, fibers, and rich source of other components with nutritional and health benefits. For the Slovenian production area, common bean is known as a vegetable crop, as a vegetable of increasing agronomic interest. So worldwide is mostly known as a crop. So it's in Slovenia, it's still as a vegetable. So at KIS, at our institute, our recently developed varieties are coming from targeted hand cross-pollination with the aim to introduce the genes which are associated with most important traits of interest regarding breeding objectives. So we know that common bean has different growth habits. So it's dwarf type and it's climbing one, and we are using it for the fresh pot consumption, and we are using for the dry seeds. So there, regarding to this, let me say, specialty of common bean plant and reproductive biology, also the breeding objectives in our breeding program are associated with that. It is very important to know that a common bean is self-pollinated annual plant with a relatively small genome. It's diploid species, and it has available reference genome since 2014. Next slide, please, Teresa. Okay. So now we are going to the breeding objectives. So for those who were attended into the module 2, this breeding program was also presented in module 3. 2 weeks ago, and the breeding objectives of the common bean in our case, are mostly orientated into the tolerance to biotic and abiotic stresses. So in the terms of biotic stress, we are talking about being common necrosis mosaic virus and anthracnose resistance, and as well as buhit resistance. And from the part of the abiotic stresses, we are focusing on the temperature tolerance and water tolerance. And of course, earliness is very important for us. And in our breeding program, we are really keen to develop varieties which are early, which prevent the plant to flower at the really hot summer temperatures, which is then the consequence that the flowers drops out of the plants and there is no yield at the end. So earliness is very important. And we are tending to develop the varieties which are in terms of snap beans for the fresh pot consumption in the type of maslinets. So we are calling this maslinets. This is a really climbing bean which is yellow with yellow pods, long pods, flat pods, and stringless pods. So it's really long, yellow, and stringless pods, and very refreshing and very good for the fresh pot consumption. And of course, for the application of the machinery during the cultivation of the common bean, it is very important that we have a favorable position of the pots on the plant. So this is relevant for the dwarf type of common bean, and of course high yielding varieties and the varieties with the favorable nutritional composition. So higher levels of proteins, iron, zinc, and lower levels of anti-nutritive components like phytic acid. So why I'm explaining you these breeding objectives? Because these breeding objectives are strongly associated and related with the traits of interest, which want to improve it during the breeding programs. And the traits of improvement are also the key traits which are followed by marker-assisted selection, so that we can identify and select loci in the genome of common bean which are associated and linked with the traits of interest which are associated with the breeding objectives. So this is the whole chain of the importance why we are talking about traits. First, breeding objective traits, and then markers, trait-related markers. Okay, so now we're going to the selection of the DNA markers. So in the morning units, and as well Teresa mentioned that the selection of DNA markers is very important. And in our case, I will now just explain how it is done, it was done in our case. So in our case, the selection was done with the markers was done with the support of our national applicative project, which was also a kind of allow us to complement the contents of the common bean breeding program, which is in Slovenia financed by our agricultural ministry, but then there is no enough funds for this basic knowledge to generate basic knowledge how to introduce it into the breeding program. So within this project, we kind of identify and select the panel of the markers which would be suitable for common bean breeding program. Okay, so how did we do it? Okay, so in our case, the markers were selected via literature survey, considering the main following issues. So in our case, the markers were already available in the literature, were already published. We just do the literature survey according to different requirements we have for our breeding program. And the first requirement is that the markers were or are species specific, so they are specific to the common bean genome. And the second one, very important thing, which was already mentioned, is that they are covering the traits of interest regarding to breeding objectives with the background information about the trait inheritance, relations, and specificity. So what it was talking about and already mentioned by Teresa. So the specificity of the traits. So are they quantitative, qualitative? We were talking about this in the module 3 2 weeks ago, dominant, recessive, and what is the level of the heritability. So all this background is very important for the selection of the markers, and how many loci you will include in your panel of markers, that you will cover the major gene which are directly associated with the key traits of interest. And in our case, it was very important as one of the criteria for the selection from literature was that they are that those markers are proven to be highly polymorphic. And as it was also mentioned by the Teresa, that they are highly applicable in terms of repeatability. So they are repeatable, reliable, quick, and cost effective. And since at our institute we have also a very nicely equipped genetics laboratory, so it's molecular lab, and we were also focused that all the analysis can be done quickly and cost efficiently in-house. So this is very important. Since we have to screen a lot of breeding material at the same time, it's very important that you will see later that we have a little automatization as well in our lab, and to a kind of boost and to do it quick and cost effective. As it was mentioned in our case, for the selection of the markers, the panel, which was pre-selected from the literature, was then optimized, tested, and validated in the core collection of common bean. So it was tested on 77 genotypes, which were actually a part of the core collection. But this core collection was actually developed from the whole collection out of 782 different genotypes coming from 12 different countries among Europe, so different geographic origin, so that we can cover the highly diverse germplasm. So at the end, we have established a panel of 24 functional trait-related DNA markers, which can be routinely used in our common bean breeding program. Hey, Teresa, please, next slide. Okay. So those are 24 markers are different of a different type. So they are SSRs, so genomic SSRs, CAPs, locus-specific PCR markers, SCARs, and as it was also mentioned in the intro part, there were also for binary and codominant genotyping. So they're both for codominant and dominant genotyping. So first, with which traits, so the panel of those 24 markers, which traits are associated with? First is gene pool determination. So as we know, for the common bean is very important that it comes from two different gene pools, from the Andean and Mesoamerican, and as well a subgroup of those two, or mixed group of those two. So since we are using in our breeding program, we are using the targeted cross-pollination, so, and the parents can be from Andean and from Mesoamerican, it is good to know the origin of our breeding line at the end. So it's very important that we know the origin. And secondly, so with this gene pool determination, we have 4 different markers. And then we are continued with 5 markers which are associated with bean common necrosis mosaic virus resistance and earliness and yield. And then we have 1 marker for bean rust resistance. Then we have 4 markers for anthracnose resistance, 2 markers for angular leaf spot, 1 marker for bruchid resistance, which is indirectly associated with the trypsin inhibitor content. Then we have 3 markers which are associated with the iron and zinc and yield related markers. And then we have two markers which are associated with the favorable root morphology and phosphorus uptake, and one marker which is associated with the gene of the major effect for the drought stress resistance. So this is our panel of the markers which are routinely used in actually every year for our breeding lines. Okay, next slide please, Teresa. So now we have selected the markers. We have a panel of the markers which is proven to be okay. So it's tested, it's validated, it's optimized, and it works well for us in the lab. So now the next question is, when applied marker assisted selection, in which stage of the breeding process? So this scheme was also presented in module 3 for the common bean breeding program, and it is very important to know that marker assisted selection is applied in filial 2 or filial 3 generation in our breeding process. So it's very important that it is applied where the segregating diversity is the highest. So in F2 generation or F3 generation is actually the highest segregating diversity. And what is also very important is, which also Teresa exposed, that this is not just a marker assisted selection as itself. So it's complementary to phenotypic selection. So what we see with our eyes on the field, the traits of interest which are expressed on the plant, and we see it with our eyes. So the plant is super, but we don't know its genetic background. So if the breeding line which is favorable for us is promising for what we see, but we don't know the genetic background, if there are genes for the resistance to bean necrosis mosaic virus, to other pathogens. And it is very important that we screen and to see if the genes for this resistance are present. And in this case, it is very important that is just to screen to see what it is within the genome. We do not rearrange the genome or do anything in the genome, but we just see the loci, if they are present or not present in the genome for those traits. Okay, and that's why it's very important that also Teresa exposed that is also suitable for the organic breeding. And the second point is that marker assisted selection is not destroying the plants at the end, because for this application we need just a small amount of the leaf tissue to do it in the F2 generation. So we do the sampling on the field, so after the phenotypic selection or during the vegetation on the plants, and continue with the work in the lab. Next slide, please. As I mentioned, so we just need for the marker assisted selection, our template is 60 milligrams to 100 milligrams of the healthy plant tissue to be sampled on the field from the F2 plants. And this is enough. And this is enough for the application of the marker assisted selection routine use set, which was explained earlier. When this is very technical part, I would say, but for those who are familiar or for those who are not familiar with this, I would rather explain it because it's very interesting to then understand the next steps. So we do the sampling, we do the homogenization of the plant sample, and then we do the extraction of the genomic DNA. As it was mentioned earlier, so we have to screen a lot of breeding materials at the same time. And since at our institute there is not just only the common bean breeding programs which is held at our institute, but there is also a buckwheat breeding program, potato breeding program, and also marker assisted selection is applied. So we have to kind of adjust our methods and reduce the costs with the automatization of our processes in the lab. Since our lab is still mainly research lab, but we have some robots as well in our lab to help us to speed up those processes as well. So first one is the robot for the DNA extraction, so for the extraction of the nucleic acids. which is MagMax. So it's very high throughput extraction method, actually, which is suitable for the extraction of the DNA for common bean. Then we check the quality and the quantity of the extracted DNA, which is then the template for the PCR. So we are using the fast PCR protocols, which are quicker for the regarding to the specific requirements for the each DNA marker which is applied for the marker assisted selection on our breeding material. And then after that we have the visualization of the results. So here have two points. First one is the binary scoring for dominant markers. So we are using it on the high resolution agarose gel electrophoresis, and we are using KeyXL as well. So here it's just the length determination, and the fluorescent labeling is not needed. But for the codominant scoring, so for the SSR markers, we are at the end generating the allelic matrix. When then here we do the fragment reaction following with the fragment analysis, which is done at the sequencer, and then reading the electropherograms to get the final allele lengths at the end for the as input data for the softwares of the population genetics, which are used at the end for the analysis of the molecular data and genotyping results. So after that, it is possible for us to highlight the most promising genotypes with the best genetic background according to the traits of interest. Next slide, please. Okay, so now just a little bit about the more in details about the analysis of the results. I would really not go into the really details, but just to have an idea and the information, how does it look like? Okay, for the binary scoring, the presence or absence of the band at the specific loci, meaning to identify the genotypes with the presence of the alleles or multiple alleles or the genes associated with the trait of interest. So it's very can be scored very quickly. But for the results of the combining then the results from the codominant genotype for the dominant genotyping and codominant genotyping, we are using the population genetics software. So for those who are familiar with the population genetics analysis, you are using, I guess, the same softwares as well. So with genealogy, structure harvester, genetics, Arlequin populations, tree view. So these are some of them are very old, but still useful since they have implemented algorithms which are still using in the R environment. So all of those are now combined with the R software and different packages for the population genetics in R. So this all allows us to evaluate the genetic diversity, the variability, the genetic structure, clustering, which is very important, the marker informativeness, genetic distances between our breeding lines, level of genetic distinguishing between breeding lines, due to different population genetic parameters. So these population genetic parameters are giving us the information about the all those variability which was explained. So genetic structure, clustering, and everything else. So it's expected heterozygosity, polymorphic information context, content, number of effective alleles. So analysis of results of analysis of molecular variability, principal coordinate analysis, allelic patterns, then UPA-gamma-A clustering in combination with the NAIS genetic distance, then the for the genetic structure plots for the genetic group. So this all combination of those parameters is very important then to interpret the results of the genetic analysis which was obtained from our genetic analysis. Next slide, please. Okay, and finally, we are about to choose. So we are about to choose to select the most promising F2 breeding lines with respect to the best genetic background according to the results of the genetic analysis. And it was already mentioned then, After that, so since it is done in F2 generation, we can continue. This allows us to continue with the phenotypic selection during the subsequent generation of selfing to make the trait stable and homogeneous, and to reduce the number of the breeding lines which are not suitable, which have no, I don't know, resistant genes, the traits of interest which are important for our breeding program. So the combination of phenotypic selection before F2 and in F2, and in combination with marker-assisted selection, we can go then further with the decreased number of the breeding lines, which are the most promising one, and has the favorable genetic background and the favorable traits of interest, which are visually described or visually expressed on the plant. And this allows us to breed more effectively during the process, maintaining the best breeding lines only with a favorable genetic background of the trait of interest. Next slide, please. So in the morning or in the previous session today, it was nicely explained that what is actually behind the discovering trait-related markers. So this here I was talking about the markers which were selected by the literature, from the literature, and somebody already did a hard work to discover, to identify, to characterize and isolate the markers and publish them. Okay, but behind that, so before that, of course, the discovery is very important, especially now which we have, let us say, modern approaches like NGS-based applications. And in our case, I wanted to put this also on the one of the last slides of for Common Bean, which was published from our side, is how do we actually discover and identify SNPs which are associated with the seed pigmentation on the common bean within the composite population. This was done by our PhD student Eva Plesteniak. And it's actually from her part of work, from her PhD thesis, and locate those SNPs in the common bean genome, and those locations and SNPs could be then used further and implemented into the breeding program as well, since the color of the seeds and the seed coat pattern is also very important for the consumers when they buy seeds, especially for the dry use. Okay, next slide, please. So this was just for the orientation. And now just to wrap up, why marker-assisted selection is important in breeding. So to refresh our memory into and to summarize what we have learned in this section. So enhanced disease resistance. So molecular breeding and marker-assisted selection allow us the precise identification and incorporation of genes which are responsible for resistance to various diseases. This leads to the development of the common bean varieties that are more resilient, to thereby reducing crop losses and improving yield stability. And as it was already mentioned today, accelerate breeding processes. So traditional breeding methods, which are allowed in organic breeding, can be time-consuming and labor-intensive. Mass significantly speed up the breeding process by enabling the selection of desirable traits at the seedling stage, rather than waiting for the plants to mature. This efficiency allows breeders to develop new improved varieties more quickly, and more quickly in the process of the common bean breeding process as well, so that we can decrease the number of the promising breeding lines which can be lead further for selfing and selection. And improved trait selection. Mass-assisted selection facilitates the selection of complex traits that are difficult to measure phenotypically, such as drought tolerance and nutrient use efficiency. And it was also mentioned today. By using molecular markers linked to these traits, breeders can more effectively select and combine the desirable characteristics, leading to the development of common bean varieties that are better adapted to diverse environmental conditions. So in those conditions which are really changed during the climate changes, which are recently present in our world. So this is actually the main challenge for all the breeders, I think, for the all plant species. So yes, it is very important that we have some tools which help us to accelerate breeding and to breed more effectively. So, and this marker sister selection surely is. Okay. Think that it is now time for Teresa again. So we will stay with the legumes. Please, Teresa, I will leave the floor to you and to the application case about the organic white looping breeding program in Switzerland.
Thank you, Barbara. So yes, from common beans to lupin beans, in this specific case, a white lupin species, the Lupinus albus, as it is an underutilized crop. I intend to shortly present it. It gained the name of the soybean of the north for several reasons. It has a very high concentration of protein in the grain, in average 35%, but can be up to 40%. And it has the best amino acid composition in terms also of the important ones for human nutrition just after soybean in the plant-based type of protein, of course. It is an excellent nitrogen fixator. So while with the soybean, after a cycle, one might come to 0, with the lupin, you definitely leave something and something consistent in the soil. It has very deep roots, which improve the soil structure. And thanks to this special type of roots, which are called cluster roots, when in the soil there isn't enough of phosphorus, these roots are produced in order to mobilize the available phosphorus. It has big, beautiful flowers, which even though they don't contain nectar, they are a good source of pollen, especially in Central European conditions when they flower in a moment where basically almost no other crop is flowering in the fields. It is a much more frost tolerant summer crop compared to the soybean, so it can be sown in Central European conditions already in March, while for the soybean one has to wait much more. And it has a good drought tolerance, so it is basically a fantastic crop for organic farming and for sustainable farming. Yet of course, it has also some challenges if we are doing breeding on this crop. It is meant to be mostly self-pollinating crop, yet there is up to 15% of cross-pollination, especially in organic fields. So the isolation from the insects is really needed in the breeding program, which of course is an additional effort in time and in management of the breeding nursery. With all the positive aspects of the crop, it comes also reasons why it is not yet much more used by the farmers. First one is that in conditions where, especially at flowering time, it can be wet but still hot, the disease anthracnose can be really damaging, even giving a complete yield loss. So the first, most important breeding target in our breeding program is resistance to this disease. The disease, it's proven by a few studies on this topic to be a quantitative resistance, and until now no gene for gene resistance type, and especially very few resistant sources have been identified, even though a screening really of genetic resources from worldwide has been done by several groups in the world. The other let's call it issue of this amazing crop is that in defense to the predators, the plant produces alkaloids in the green part, which are translocated then in the grain. The quinolizidine alkaloids, they are bitter but also toxic. So there are suggested threshold for the use in food, which is 200 milligram per kilogram, and for the use for feed for the animals, 500 milligram per kilogram. The wild type have an incredible amount of alkaloids in the grain. Yet there are most of the varieties, which are modern varieties, which are the ones used in Central European conditions, they are so-called sweet variety. Surely they are not sweet, but we mean with that the lack of alkaloid accumulation, or the accumulation of very little amount. As it's such an important component for the plant, they don't exist lupin plants without alkaloids, but you can have varieties, genotypes, which accumulate a very low amount, hopefully below the threshold for the use. This trait is controlled by several recessive loci, of which one major locus, the pauper, was in 2023 published with the causal mutation published, making it possible to do marker-assisted selection. Our breeding program initiated 10 years ago by my colleague and responsible breeder at FiBL, Christine Ankern, and which is in collaboration between our research institute FiBL and the independent breeding organization GZPK in Switzerland, is a fully organic breeding program. The crossings are usually trying to cross resistance, which are usually bit late because they are genetic resources, with elite material, which is with low alkaloid and early ripening. And then all the selection, the phenotypic selection, is done in certified organic fields, 2 in Switzerland and 1 in Germany. Since 2018, thanks to the contribute of 2 colleagues, who, one Joris, who finished, and the other Andras, who is currently doing his PhD on this topic, we are trying to integrate marker-assisted selection in our breeding program. The issue, in order to be able to do so in this crop for our trait of interest, is that first of all we need to advance with the molecular tools, because unlike for the case that Barbara presented, there are basically no validated markers for this crop that we can use. So we first have also to do the discovery and the validation in order to be able to implement them. And then be able to integrate with the, let's say, the limitation and the trade-off of being a small independent breeding program. So with the resources, with the limited type of resources we can have. Yet it is an underutilized crop, but we do have the reference genome and 39 resequenced accessions, and there are a few groups in Europe that are joining efforts in the breeding of this crop. So related to our question with the alkaloids, there are at least 5 different genes known from, especially from the older times of the first breeding attempts in the in this crop. The one which is quite widely deployed in the modern varieties is the so-called pauper, for which the causal mutation was published in 2023. And there is others which decrease the reduction. It's not as strong as pauper, which are basically not really the region in the genome which determines this this reduction is basically not known. Our question is that often even the varieties which carry the pauper mutation, they when harvested, the grain, unfortunately, it's above the limit, the suggested limit for human and animal consumption. So we would like to develop varieties which have a very low and very stable across years and environment amount of alkaloids. And the way we would like to do that, it's by stacking two different low alkaloid determinants. So very good for us. In 2023, the paper was published, yet in order to be able to have a stacking of two different ones, we would still need to have a second one. So we first had to be able to determine a second QTL for low alkaloid determination, and then to verify if combining the two in a breeding line really determines a drop in the amount of alkaloid accumulated in the grain, and then be able to use it in our breeding program. So the material that we used is segregating population from our breeding program, a crossing between pauper, you see here sweet recessive small p, and dieta, which is also a modern variety, sweet for an unknown determinant. We had a crossing, the F1, which of course was bitter, and then in 2023 we had a segregating F2 population, which as of hypothesis, it had a segregation sweet to bitter 7 to 9, so we could hypothesize that there would be a second determinant beside pauper acting for the level of alkaloids in this population. We applied bulk segregant analysis. We phenotyped in a rough way for alkaloid content all the F2 plants, and we applied bulk segregant analysis. The way we did that is that via our scoring, we had the segregation of our population from very sweet, you see this score 4, to very bitter, and this is in the all F2 population. Luckily, we could genotype for pauper, so we could identify which plants in the F2 would be sweet because they are homozygote for the positive allele on pauper. We eliminated this from our study, and this is the distribution of the other part of the population. So you see that the lowest value, it's a bit higher because pauper is the determinant which gives the strongest reduction, yet we could identify a bulk of sweet genotypes and a bulk of bitter genotypes, which were sequenced together with the parentals, with the bulks of two parentals, to respond via bulk segregant analysis to the question if there are genomic regions which are monomorphic within the sweet or the bitter bulk, but that are polymorphic between the two bulks, so that we could infer QTL related to our trait of interest. The bulk segregant analysis was successful, so we could identify a new QTL on chromosome 5. From this step, then we continued basically with the approach that I was describing in the introduction. We selected a number of SNP markers in the QTL region, we developed PACE assays, which is basically CASP markers, so PCR-based marker to be able to genotype the SNP of interest in the with qPCR in our genotypes. We did validate these markers in a diversity panel, in this case of 34 accessions. We selected a descriptive marker, which could be used in our then F3 population to identify individuals which have stacked the two alleles from the pauper and from the newly determined QTL. We then phenotyped this, and we could verify that actually the alkaloid value is very low, so this could prove our hypothesis. Where do we stand now? We stand now that having breeding lines which are very low in alkaloid is not useful unless they are also resistant in anthracnose, at least for our growing condition where anthracnose is very damaging and very present. So the next step is to combine the mass for low alkaloid with the selection phenotypic and genotypic for anthracnose resistance. The anthracnose resistance is a much harder trait to tackle because it is quantitative, and because, as I said, there is very few lines or genetic resources with resistance which were identified that can be used to improve the breeding material. Yet, we could use 3 loci from literature from the first QTL study on this trait. Our colleague Joris in 2022 published GIVAS, where we could identify other 3 SNPs. Unfortunately, we were not really able to validate those ones in a validation panel. So we followed up with a different approach by using the same data set for our GVAS in order to do genomic prediction, followed by marker selection. So the approach, the simplification approach that Michael was explaining in the morning, in order to have 50 loci to use for, let's say, reduced genomic selection model. Additionally, thanks to our collaboration with LFL, another institute in Germany that works also on white lupin breeding, we could publish GVAS and genomic selection based on another panel and based on field phenotyping. And these are new marker trait associations which we can try basically to implement in simplified, reduced genomic prediction model. So, of course, the genotyping of the accessions that were part of the 2 GVAS panels, they were in one case genotyped by GBS and in the other case by VGS. So we have a very huge number of markers, but it is not economically feasible to apply genomic selection with this high number of markers for us. So we proceeded with marker selection in order to keep a good predictive ability and to have a number of markers which is affordable for us to genotype. In this case, we have chosen biomark X array, which allows to genotype in one go 96 markers by 96 accessions, so it's cheap and high throughput. And together with our collaborators, the LFL, the CREA in Italy, and the GZPK in a European project, we are trying to bring this from the conceptual phase to the application phase of marker-assisted selection. So we do still have a few steps towards the full implementation in our breeding program. Very well. I think we are in perfect time with our agenda to continue with a short quiz. Alexandra, thanks for your question. So Alexandra asks, I said a couple of times, underutilized and neglected crop for the lupin. So she wonders how small is the acreage of this crop in Switzerland. By the statistics of the last 3-4 years, and it's relatively stable. It is about 400 hectares. And this includes not only the white lupin, but includes also the blue lupin, the narrow-leafed lupin, and it's both organic and conventional. So it is a small niche crop with a lot of potential for the future, and especially interest by some food processors, for example, for vegan milk alternatives, or in Switzerland there are some companies that produce alternative to coffee, so lupin coffee. So we hope that the interest from the food processing side and hopefully in the near future coming in of new varieties which have solved the anthracnose issue or reduced that can increase the cultivation. And the second part, it's about the alkaloids. And if the reducing the alkaloid content, it has an impact on the plant growth. Yes. Normally, the plants with very reduced content of alkaloid are less vigorous and they accumulate less biomass compared to the wild type. So it is, how to say, the task of the breeder to be able to select plants which have a good architecture, a good biomass, and still a good weed suppression, especially in organic, and still be among the sweet ones. And there is variability in order to do this type of compromise.
I guess, Teresa, there is one question for common bean.
Yeah.
And Dario, thank you for your question. So can you tell us the cost of marker-assisted selection and all breeding costs for develop one bean variety in KISS? This one is really, I cannot answer to this question at the moment because the final calculation according to the so considering all the steps of the breeding process, including the marker-assisted selection, is updated now, and it is actually under work. So it will be done, I think, in very short time, but at the moment I don't have a final, let me say, a final number. How does it costs to develop one variety, including all the costs. So for this, I cannot answer at the moment.
I would add that it's also a very difficult calculation to do, as also the single variety comes from a breeding program. So the cost of the first variety is definitely different of then set and ongoing breeding program. And also it depends on very much on how much synergy is done between the research and the breeding part. Barbara, when you will be able to do some rough estimation for your breeding program, I guess that will be of a very good inspiration for other small size independent breeding organizations.
Yeah, yes, we have evaluated, financially evaluated some parts of the common bean breeding process, but not to do the complex and the whole evaluation for the whole process. And as you said, I completely agree with the statement that it's different. How does it cost if you have the first variety and all the following ones, which is very different. So in our case at KISS, as I've mentioned, so it's a public breeding program which is financed by our ministry, agricultural ministry, but also we all the basic knowledge is also from the different research projects, national ones, international ones, and then this knowledge can be directly implemented into the breeding program. So that's why it's even harder to financially evaluate the whole process.
We are asked if we will share the presentation. Yes, for sure. All the recordings and the presentations of the different modules and units will be published as soon as edited on the LiveSeeding website. I guess that you still see the presentation in the screen. I have a last slide, which is the homework. The activity that we planned with Barbara is to ask you to present an example. As there are some of you that already work with market assisted selection, it would be great to get a summary of the real example of how you apply market assisted selection in your work. And if you are not, just a potential case from literature, so you can do a literature research for that. The way to respond to us is via short questionnaire. We will get the information of who you are just in order to compile it with the other homework for the certificates. And we would like that you pick a crop of interest and the target traits for which one can apply marker assisted selection, the type or types of markers and the number for each to provide us the references, for example, the scientific publications from where this marker was, how this marker was picked in order to be used in marker assisted selection, and to provide a short description of the protocol in your example case. And we would be very glad if you can respond to the survey